Hello everyone, welcome to our own YouTube channel Shantanu's Bioprep LifeScience. In the previous lectures we have discussed glycolysis, regulation of glycolysis, and the first lecture was introduction to metabolism. In this lecture we are going to discuss in detail the process of pyruvate oxidation. Okay. Pyruvate oxidation is the connecting link between glycolysis and Krebs cycle. How is it connecting link? In glycolysis, we know that glucose is oxidized and converted into pyruvate. In Krebs cycle, we know that acetyl coenzyme A combined with oxaloacetate to form citrate. So acetyl coenzyme A is required. In pyruvate oxidation, pyruvate is converted into acetyl coenzyme A, and this acetyl coenzyme A will enter in the Krebs cycle. So can we observe that pyruvate oxidation is the connecting link between glycolysis and Krebs cycle? Okay. Location of pyruvate oxidation is in mitochondrial matrix in the case of eukaryotes, because membrane bound cell organelles are present in eukaryotes, whereas in prokaryote it will occur in cytosol, because in prokaryotes membrane bound cell organelles are absent. Okay. Pyruvate oxidation occurs in the presence of oxygen, so you should remember this point. Okay. It is an irreversible reaction because the change in free energy for this reaction is large negative, due to which the chemical reaction will occur in forward direction and not in backward direction. It is an exergonic process because during this reaction heat will be released. Okay. As we can observe that pyruvate will combine with coenzyme A, which will be catalyzed by pyruvate dehydrogenase complex, and it will get converted into acetyl coenzyme A with the liberation of CO2. Pyruvate dehydrogenase complex will have cofactor NAD positive in oxidized state, which will get converted into reduced state, that is NADH plus H plus. We will understand this in more depth. So as we can observe the pyruvate molecule, there will be decarboxylation, that is removal of CO2, as shown by the black dotted lines. And upon or in the presence of coenzyme A, in the presence of pyruvate dehydrogenase complex, it will get converted into acetyl coenzyme A. Okay. So this CO2 will be released as shown here. The remaining will combine with this coenzyme COAS, and I have shown it over here. COAS will combine with this remaining 2 carbon, and it will form acetyl coenzyme A. The H from this coenzyme A will be accepted by this oxidized state, that is NAD positive, and it will get reduced to NADH plus H plus. We know that loss of hydrogen atom and electron is known as oxidation. Acceptance of hydrogen atom and electron is known as reduction. So the hydrogen atom from the coenzyme A will be accepted by oxidized NAD plus, and it will get converted into reduced NADH plus H plus. Pyruvate oxidation step is known as oxidative decarboxylation. Why? Oxidation is going to occur because hydrogen atom from coenzyme A is getting lost, and decarboxylation because this CO2 group from pyruvate is getting lost. Oxidative decarboxylation. Okay, easy. Next, pyruvate dehydrogenase complex is a multi-enzyme complex because it has 3 individual enzymes, E1, E2, E3. So it is composed of 3 individual enzymes. E1 enzyme is known as pyruvate dehydrogenase. Number of polypeptides present are 24. Cofactors are thymine pyrophosphate TPP. Function is decarboxylation and oxidation of pyruvate. We will understand these function in next slide. Enzyme 2 is known as dihydrolipoyl transacetylase. Number of polypeptides present are 24 and the cofactors are lipoic acid and coenzyme A. Its function is the formation of acetyl lipomide from hydroxyethyl TPP and transfer of acetyl group to coenzyme A. E3 enzyme is known as hydrolipoyl dehydrogenase. Number of polypeptides present are 12. Cofactors are NAD plus and FAD. Function is reoxidation of dihydrolipoamide. Now we will understand the mechanism of pyruvate dehydrogenase complex in detail in the next slide. So I think you are not able to see what is there on the slide because of my video. Let me hide the video. Okay. So I think it is visible now. Okay. First statement is enzyme 1 will catalyze the decarboxylation of pyruvate and forms hydroxyethyl TPP. So I have shown pyruvate molecule over here. Enzyme 1 will catalyze decarboxylation of pyruvate. So this CO2 is going to liberate. Okay. So decarboxylation has occurred. So this group which is left will be combined with TPP. Enzyme 1 has TPP as a cofactor. This TPP will combine with whatever that is left after the removal of CO2 and it will form hydroxyethyl TPP. As the name suggests, hydroxy means it will have OH group and rest is same as what is present over here. Okay. And this hydrogen is because carbon atom form 4 bonds which we have discussed in the previous lecture. Because if we would have written it like this, then carbon was going to form 3 bonds over here and 1 bond over here. So total 4 bonds. But for this carbon, it will be only 3 bonds. 1, 2 and 3. Which is wrong thing. Because we know that carbon atom has to form 4 bonds. So if we write hydrogen over here, we will observe that it will form 4 bonds. By this trick also you can eliminate incorrect structures given in the options. If a question based on structure is asked, by this trick you can also eliminate incorrect option even if you don't know the structures. You have to remember that carbon atom will form 4 bonds. So now you can observe that this carbon atom will form 1, 2, 3, 4 bonds and this carbon atom will also form 4 bonds. 1, 2, 3 and 4. So now you understood why H is there. Okay. So this TPP will again go and bind with enzyme 1, so that next cycle of pyruvate oxidation can occur. After next pyruvate decarboxylation step, whatever is left can be accepted by TPP. So TPP will be returned to enzyme 1. Okay. So now this group is left. This entire group is left. Okay. The next statement is E1 enzyme will catalyze oxidation of hydroxyethyl group, which results in the acetyl group formation. Electrons from this oxidation will then reduce the disulfide of lipoamide, which is bound to enzyme E2. Now from this hydroxyethyl group, this hydrogen will be released by oxidation, because we know that oxidation is loss of hydrogen atom and electron. Reduction is acceptance of hydrogen atoms and electron. So after this hydrogen atom gets lost, this hydroxyethyl group will be converted into acetyl group. Okay, it will get converted into acetyl group. Now there is one more thing. Oxidized lipoamide is present, or it is bound to enzyme 2. Okay, but when the oxidized lipoamide will accept this hydrogen, when the oxidized lipoamide will accept this hydrogen, it will get reduced, because we know that acceptance of hydrogen and electron is reduction. So this oxidized lipoamide will get converted into reduced lipoamide, and this acetyl group will get transferred to reduced lipoamide, and it will form acetyl lipoamide. As simple as that. Acetyl group is transferred to reduced lipoamide and it will get converted into acetyl lipoamide. Okay. Next step. Enzyme 2 catalyzes transfer of acetyl group to reduced lipoamide, which forms acetyl lipoamide, which we have already discussed. Then enzyme 2 will catalyze the transfer of acetyl group to coenzyme A. Okay. So this acetyl group will be transferred to coenzyme A. Acetyl group plus coenzyme A will form acetyl coenzyme A. Okay. And who is going to catalyze it? Enzyme 2. It will catalyze the transfer of acetyl group and transfer it to coenzyme A. So acetyl plus coenzyme A will give acetyl coenzyme A. So what is left now? Reduced lipoamide. So this is reduced lipoamide. Okay. Next step. Enzyme 3 will catalyze reoxidation of reduced lipoamide. Electrons are first transferred to FAD and then to NAD plus. Okay. So now acetyl coenzyme A is formed. But this reduced lipoamide should be converted into oxidized lipoamide so that next pyruvate oxidation cycle can continue. Because when this step will continue, then this step will continue, and when there is transfer of acetyl group to lipoamide, we need oxidized lipoamide, without which this step cannot occur. So this reduced lipoamide should be converted into oxidized lipoamide so that next cycle of pyruvate oxidation can occur. And this reoxidation of reduced lipoamide is catalyzed by an enzyme E3. So this is very important. Enzyme 3 catalyzes reoxidation of lipoamide and not enzyme E2. Okay. Reduced means acceptance of hydrogen atoms. That's why this is known as reduced state. So if this reduced state has to be converted into oxidized state, what should we do, or what should be done? We have to remove these hydrogen atoms from this reduced lipoamide. We need to remove the hydrogen atoms. Okay. So these hydrogen atoms will be accepted by FAD, which is the cofactor of enzyme 3. FAD is in oxidized state, when it will accept 2 hydrogen atoms, it will get converted into reduced state, that is FADH2. Because we know that in oxidation means loss of hydrogen atoms and electron, reduction means acceptance of hydrogen atoms and electron. Okay, so FADH2 now has 2 hydrogen atoms, and we know that in one hydrogen atom there is 1 proton and 1 electron, and therefore the net charge is 0. Okay, now this FADH2 has to be converted into oxidized state so that next cycle of pyruvate oxidation can occur, so that this step will occur, this step will occur, this step will occur. Reduced lipoamide will be transferred, or sorry, reduced lipoamide will be oxidized, and then when the hydrogen atoms which are released should be accepted by oxidized FAD. Yes, therefore we need to convert this reduced FADH2 into FAD so that next pyruvate oxidation cycle can occur. So these hydrogen atoms will be accepted by next cofactor of enzyme 3, that is NAD+, which is present in the oxidized state. Its charge is positive because we have discussed this in previous lectures in very much detail. It is positive charge because any substance from where there is loss of electrons, that is loss of negative thing, it will render the molecule as positively charged. Okay, so we write FAD form, sorry, we write reduced form of FAD as FADH2, but we write reduced form of NAD+ as NADH plus H+. This also we have discussed in the previous lecture. So students who are directly watching this video should also watch previous videos so that all your basics will be clear. So we write this as NADH plus H+ and not NADH2 because, so if I write 2 hydrogen atoms over here, and we know that in one hydrogen atom there is 1 H+ and 1 electron minus. So in 2 hydrogen atoms I have written 1 H+ plus 1 electron. It should be noted that when the NAD+ will accept hydrogen, it will accept 1 hydride ion. Hydride ion is 1 H+ and 2 electron. This is hydride ion. This we have discussed in previous lecture. So this NAD+ will accept 1 hydride ion. So if we see, this thing will be accepted as a hydride ion. Yes, and what is left? This thing is left. This free hydrogen ion or free proton will be accepted as it is. So NAD+, that is oxidized state, will accept 1 hydride ion and 1 proton or 1 hydrogen ion as it is. Okay, and this is why we write NADH plus H+ and not NADH2. Okay, okay. So this NAD+ will get converted into NADH plus H+. This NADH molecule will enter electron transport chain and result in the formation of 3 ATP. How this will occur, we will discuss it when we will study electron transport chain in detail. So when one NADH molecule passes through electron transport chain, it will result in the formation of 3 ATPs. Okay. So this was the mechanism of pyruvate dehydrogenase complex. Now we will quickly summarize all these steps. Okay. Okay. So this is a pyruvate molecule which will undergo decarboxylation, which will be catalyzed by enzyme 1. So CO2 group will be removed from pyruvate. Okay. Enzyme 1 has TPP as a cofactor. The group which is left after the removal of CO2 from pyruvate will combine with TPP and form hydroxyethyl TPP. Then there will be oxidation of this H from hydroxyethyl TPP, due to which it will get converted into acetyl group. Okay. This TPP will again detach and get combined with enzyme 1 because TPP is a cofactor of enzyme 1. So this acetyl group which is left will combine with lipomide. How it will occur? Enzyme 2 has bound lipoamide. Okay, it has bound lipoamide which is initially present in its oxidized state. Okay, due to the oxidation of hydroxyethyl TPP or hydroxyethyl TPP, in oxidation hydrogen ions and electrons will get released, due to which when it will get accepted by oxidized lipomide, we know that upon the acceptance of hydrogen atom and electron, the molecule will get reduced. So this oxidized lipomide will get reduced as shown over here. This SH means it is in reduced state now because acceptance of hydrogen atom has occurred. So this oxidized lipomide is in reduced state now. This acetyl group which is formed after the removal of this H will get or will combine with the reduced lipomide and it will form acetyl lipomide. Okay, it is easy. Now this reduced lipomide will separate from this acetyl group. Okay, as shown over here. So this is reduced lipomide and the acetyl group will combine with coenzyme A to form acetyl coenzyme A. Acetyl group plus coenzyme A will form acetyl coenzyme A, and the group which is left is reduced lipomide with 2 hydrogen atoms as shown over here, SH. Okay. Now there should be reoxidation of this reduced lipomide to oxidized lipomide so that next step or next cycle of pyruvate oxidation can occur, because we know that initially it should be present in oxidized state, and only then after the oxidation of this hydroxyethyl group, the electrons and H plus ion which have been liberated will be accepted by oxidized lipomide to form reduced lipomide, and this acetyl group can combine with the reduced lipomide to form acetyl lipomide. So for this to occur, initially the lipomide should be present in oxidized state. Therefore, we need to reoxidize this reduced lipomide to oxidized lipomide. So the next cycle of pyruvate oxidation can occur. Okay. So in the reduced lipomide, the two hydrogen atoms will get accepted by the oxidized FAD, and after the acceptance of 2 hydrogen atom by FAD, it will get converted into FADH2. And one more thing I forgot to tell, reoxidation of reduced lipomide to oxidized lipomide will be catalyzed by enzyme 3 and not enzyme 2. Okay. So after the acceptance of these 2 hydrogen atoms by the oxidized FAD, it will get converted into reduced FADH2. Okay. From this, the hydrogen atoms will be liberated. Why? Because this reduced FADH2 should be again converted into oxidized FAD, so that next cycle of pyruvate oxidation can occur. Okay. And then when this reduced lipomide will actually lose the hydrogen atoms, it will get converted by FAD. because initially it was present in the oxidized state, that is FAD. So for the next cycle of pyruvate oxidation to occur, the initially FAD should be present and not FADH2. Therefore, from FADH2 we need to lose 2 hydrogen atoms, then it will get converted into FAD. These 2 hydrogen atoms will be converted, will be accepted by NAD plus, which is present in oxidized form, to get converted into NADH plus H plus. And we know that one H will be accepted as a hydride ion and one which is left is accepted as a hydrogen ion, that is H plus. This NADH molecule will enter or pass through electron transport chain to give rise to or to result in the formation of 3 ATP. Okay, so I hope it is clear now. Okay, easy. I know this is little difficult topic. You can, if you didn't understand it, you can replay the video. Okay, let us look at the inhibitor of pyruvate dehydrogenase complex. Arsenite ions and mercurite ions inhibit pyruvate dehydrogenase complex. Arsenite ions will bind to the thiol group of lipoic acid or lipoamide and lipoate. These are all 3 names of same lipoamide. Okay, and makes it unavailable to serve as a cofactor. So if this is lipoamide which has been bound to enzyme 2, it has SH group in reduced state. Okay, so if this is SH group, this black pen is SH group, and my fist is lipoamide, my hand is enzyme 2. Okay, arsenic ions will come and bind with the reduced lipoamide SH group. This complex derivative will be formed. Because of this, the hydroxyethyl TPP, when it will oxidize into acetyl group, the acetyl group cannot bind to the reduced lipoamide because arsenite ions has bound with it, due to which there will be no formation of acetyl lipoamide. If there is no formation of acetyl lipoamide, next step of pyruvate oxidation will not occur. Okay, now we will understand regulation of pyruvate dehydrogenase complex. Let me hide the video. Okay, now you can see what is written on the slide. Okay, for this to understand, you should remember 2 important points. The pyruvate dehydrogenase complex is active in unphosphorylated or dephosphorylated state, and the pyruvate dehydrogenase complex is inactive in phosphorylated state. It is inactive in phosphorylated state. PDH complex is also a bifunctional enzyme, as it plays 2 roles, kinase and phosphatase. It has 2 domains, kinase and phosphatase domain. We know that whenever a word kinase is there, it will result in it will catalyze the phosphorylation. So what it will do, it will take a phosphate group from ATP and it will add the phosphate group to PDH complex. Okay, so here we have phosphorylated the PDH complex, and we know that the PDH complex is inactive in phosphorylated state. So PDH kinase will inactivate the PDH complex. Okay, this inactive PDH will get converted into active PDH when there will be removal of phosphate group, because we know that PDH complex is active in unphosphorylated or dephosphorylated state. So this phosphate group should be removed, which will be removed by PDH phosphatase. Whenever there is a term phosphatase, we know that it will catalyze the removal of phosphate group. So it will remove the phosphate group which has been attached and convert the inactive PDH to active PDH. Okay, so this normal pathway we have understood now. Now we will study the allosteric activator and inhibitors of PDH kinase and PDH phosphatase. increased pyruvate levels, increase NAD plus and increase coenzyme A levels will inactivate PDH, or it will inhibit PDH kinase. So what is the logic behind it? We know that NAD plus will get converted into NADH plus H plus. Okay, but here NAD plus levels are more, which means the body is in energy deficient condition, because NADH will pass through it electron transport chain and give rise to ATPs. NAD plus will not pass through electron transport chain. So if NAD plus levels in the blood is more, which means cell is in ATP deficient condition. So in this situation, we need ATP. For this, pyruvate oxidation should occur, because when pyruvate oxidation will occur, pyruvate will get converted into acetyl coenzyme A. Acetyl coenzyme A will enter into Krebs cycle and produce more ATP, and which is what we want. So NAD plus will inhibit PDH kinase, and due to this PDH will be active, because we know that PDH kinase will add phosphate group to PDH complex and convert it in inactive form, which we don't want. We want active PDH complex, so that pyruvate oxidation into acetyl coenzyme A can occur and further ATP formation can occur. So we need to inhibit PDH kinase for the pyruvate oxidation to occur, and that is why NAD plus is preventing PDH kinase. Similarly, coenzyme A. If the levels of coenzyme A in the blood is high, it means coenzyme A is in free form. It is not bound with acetyl coenzyme A. If it is not bound with acetyl coenzyme A, or we can understand it like this also, we know that in pyruvate oxidation, pyruvate plus acetyl coenzyme A, pyruvate plus coenzyme A will form acetyl coenzyme A by oxidative decarboxylation, and CO2 will be released. So if this coenzyme A is present in high concentration, it will favor the formation of acetyl coenzyme A. It can simply bind with the pyruvate dehydrogenase complex enzyme. For pyruvate oxidation to occur, we need to inhibit PDH kinase enzyme, because PDH kinase enzyme is inactivating the pyruvate dehydrogenase complex. So for pyruvate oxidation to occur, inhibition of PDH kinase should occur, and this is what it is doing. And then similar role is played by pyruvate. Pyruvate is the substrate of pyruvate dehydrogenase complex. When pyruvate levels will be higher, it can bind with the pyruvate dehydrogenase complex and promotes its oxidation. So for pyruvate oxidation to occur, we should inhibit PDH kinase, because PDH kinase is going to inactivate pyruvate dehydrogenase complex. When PDH kinase will get inhibited, pyruvate oxidation can occur, and which is what pyruvate wanted. This we have also seen in the previous video. When the blood glucose levels is high, it will bind with the hexokinase enzyme, and hexokinase will catalyze the conversion of glucose to glucose 6-phosphate. So if you haven't watched that video, please watch it from lecture number 1, okay, so that you can understand the concepts and basics much clearly. Okay, now ATP, NADH and acetyl coenzyme. If ATP is already present in high amount, then there is no need of excess ATP production or excess ATP formation, because acetyl coenzyme A, which will be formed from pyruvate oxidation, will enter into Krebs cycle, and then further ATPs will be formed. But energy now is already present in high amount, then we need to stop pyruvate oxidation. For this, we need to activate PDH kinase, because PDH kinase will convert the active PDH into inactive PDH. Due to this, pyruvate oxidation will not occur, and that is why ATP will promote the activation of PDH kinase. Similarly, NADH. This NADH, if it is present in high amount, then there is ATP sufficient condition in the cell, because this NADH will pass through electron transport chain and produce more ATPs. And if NADH concentration in the blood is high, then energy is sufficient inside the cell. Then the body will try to stop excess ATP formation. For that, we need to inhibit pyruvate oxidation. So NADH will activate the PDH kinase, which will convert the active PDH into inactive PDH, due to which pyruvate oxidation will not occur, and that is what NADH wants. Similarly, acetyl coenzyme A. If acetyl coenzyme A levels in the blood will increase, it will enter more in Krebs cycle. More ATPs will be formed. But body will try to stop all those processes which will produce excess ATPs. Okay. Now in the cell, the concentration of acetyl coenzyme A is more ATPs will be formed. Then there is no need of pyruvate oxidation. There is no need of excess ATP formation. So the body will try to inhibit the pyruvate oxidation. So this acetyl coenzyme A will promote the activation of this PDH kinase. This PDH kinase will convert the active PDH to inactive PDH. Due to this, the pyruvate oxidation will not occur, and this is how excess ATP formation will not occur, which is good according to body. Okay. So this was the logic. Now we will understand this part. This calcium and magnesium ions are released during muscle contraction, and these calcium and magnesium ions will combine with PDH phosphatase and activate it. Okay. When the PDH phosphatase will get active, it will remove the phosphate group from PDH complex. When the phosphate group will be removed, it will get converted into active PDH. Okay, it will get converted into active PDH, due to which pyruvate oxidation will occur, due to which ATP formation will occur. Okay. Insulin. The role of insulin is to suppress or reduce the blood glucose levels. So if the blood glucose levels in the body is high, insulin will be secreted. It will try to reduce the blood glucose level. It will promote the glycolysis. After glycolysis, pyruvate will be formed. Pyruvate will enter pyruvate oxidation. Okay. Then again, in the next cycle, glucose will be degraded or consumed to form pyruvate. Pyruvate will again enter pyruvate oxidation. By this continuous process, we can observe that the glucose is going to be consumed, and by this, the elevated blood glucose level will start to reduce. So insulin want pyruvate oxidation to occur. So it will activate PDH phosphatase. PDH phosphatase will convert inactive PDH to active PDH, due to which pyruvate oxidation will occur, and which is what insulin wants. So this was the regulation of pyruvate dehydrogenase complex. So I think you have understood it. It is very much easy. Even if you are unable to grasp this, you can replay the video. Okay, this is the best video on YouTube. Okay, I have tried to explain it in much simpler manner. Okay, next slide. Biochemical importance of PDH. Deficiency of vitamin B1 or thiamine causes lack of TPP, which inhibits PDH activity, which results in pyruvate accumulation. If there is a deficiency of vitamin B1, then there will be deficiency of TPP, that is thiamine pyrophosphate, due to which E1 enzyme activity will get inhibited, because no one can bind with the pyruvate group after its decarboxylation, because we know that it will, after decarboxylation of pyruvate, hydroxyethyl TPP is formed. If TPP is not there, hydroxyethyl TPP will not get formed. So due to deficiency of B1 vitamin, there will be deficiency of TPP, and further pyruvate oxidation steps will not occur, due to which pyruvate will accumulate, because it will not get converted into hydroxyethyl TPP. Okay, and there is no one to catalyze the decarboxylation step also of pyruvate, because decarboxylation step is also catalyzed by enzyme E1, that is pyruvate dehydrogenase, and the cofactor of pyruvate dehydrogenase is TPP. If there is no TPP, no decarboxylation. No decarboxylation, pyruvate accumulation will occur. In vitamin B1 deficient alcoholics, this accumulated pyruvate will be rapidly converted into lactate, and this condition will be known as lactic acidosis. In inherited deficiency of PDH patients, so if there is some gene mutation due to which enzyme 1, 2, 3 of pyruvate dehydrogenase complex is not synthesized, in such case this accumulated pyruvate will also rapidly convert into lactate, which will result in lactic acidosis. The symptoms of this is reduced appetite, okay, and muscle cramps. Okay, next, so this we have already discussed. In glycolysis pathway, glucose will get converted into pyruvate. 2 molecules of pyruvate are produced in a glycolysis pathway. In pyruvate oxidation, 1 pyruvate molecule will be oxidized into acetyl coenzyme A, 1 NADH molecule will be produced, 1 NADH molecule will enter into electron transport chain to form 3 ATPs. If there are 2 pyruvate molecules which have been formed in glycolysis, these 2 pyruvate molecules will oxidize to acetyl coenzyme A. When 2 molecules of pyruvate will oxidize, 2 NADH molecule will be produced. 1 NADH molecule give rise to 3 ATPs, 2 NADH molecule will give rise to 6 ATPs when 2 NADH molecule will enter or pass through electron transport chain. So total 6 ATPs are produced through pyruvate oxidation upon oxidation of 2 pyruvate molecules. In glycolysis, 8 ATPs are formed. In pyruvate oxidation, 6 ATPs are formed. So total 14 ATPs are formed till now. In the Krebs cycle, we will discuss the remaining ATPs which will be formed, and at the end of Krebs cycle, we will calculate how many ATPs are formed from complete oxidation of glucose molecule, 1 glucose molecule. Okay, so I hope you have understood this topic. This topic is little complicated, okay, and in the reference book it is given in more complicated way. You just need to understand oxidation and reduction concept. After understanding that, you can analyze any biochemical pathway on your own. Okay, you don't even need to refer the material or study material, different study material. You can analyze the biochemical pathways on your own. You just need to remember what is oxidation and reduction. You can also easily analyze oxidative phosphorylation steps. Okay, so most of the students were scared of biochemical pathways, but now you can understand it. I think After watching my lectures, I am pretty much sure that you are able to understand the concepts which are involved. Okay, if you have understood up till now, you can easily solve previous year questions. Even BSc student can solve PhD entrance question after watching my lectures. Okay, and you need to revise the concepts. Okay, you can make a list of previous year question and try to solve it after watching my lectures. I am 100% sure that you will be able to solve the question. It can be of any level, BSc, MSc, PhD, you will be able to solve it. Okay, so I hope you liked my video. Please don't forget to like, share and subscribe to my YouTube channel, and also don't forget to press the bell icon so that you won't miss any notification when my next video gets uploaded. You can also join my Telegram group. Group name is Shantanu's Bioprep LifeScience. You can search it on Telegram. It has more than 3000 questions, quality questions, short notes, tricks, study materials, and I also upload upcoming exam updates. Okay, so you can join it if you want. Okay, see you in the next video. Thanks for watching.